We can't blame printer quality. That's if you're referring to the fading of the ink, those posters are 2 years old, I think.
Oh, really?
Because we didn't do posters last spring because of COVID.
Oh, okay.
So they'd be from the year before. So we won't hold fading posters against them. We can, if the picture is bad, we can blame the student.
Well, I would say one I put on there, like they had, like one of them specifically was bright yellow. Even if the quality was still good, you can't see white words. It's like it was just a lot coming at you.
Are we gonna do posters this year?
Yes, even if we are presenting them online, we will still print posters, and you may take a picture of yourself next to the poster.
Oh,
But there will be no university-wide poster session.
I don't understand that.
Use a screwdriver. I don't care what's that, it's just that string. You're spun.
He doesn't have concentration. He just does a general. I was like, I'm gonna roll, and then I looked down and I was like, 155. And she approved it. Oh, beautiful.
Yeah.
Is it appropriate to email your advisor if they haven't responded?
Yes.
But you emailed him last Thursday and I haven't heard anything.
Yeah, you can remind him gently. Dr. Mosey, have you had a chance to look at my paper? I'm gonna stay out of that one.
Yes, yes.
I already gave him my organic lecture for the whole year, so can't do him too many favors. Is everybody back? Let's check. Make sure you check into the app also, attendance.
So is everything just shifted down like one date, like so when all of the assignments are due?
No, they were always due on
Tuesday.
Yeah, no. Yeah, that one was due.
Oh, I went off of the syllabus.
Yeah.
We always were gonna change.
So there's a due to advisor due to Moodle.
Okay.
Okay, so the common theme that I'm seeing for poster likes and poster dislikes is that things are easy to follow. We like bullets. So if you go downstairs and look at the biology poster, they like their paragraphs downstairs. I do not like paragraphs. It's too much reading. So we like bullets, things that are easy to follow. Lots of you said pictures, visual aids, tables and graphs that are easy to follow. So these are the things that you want to think about when you're making your posters. If you have a lot of statistics or something in your results or whatever, think of a way to put that into a table or some sort of figure, because it's easier to get the data from a table than to read about it. When you're writing your thesis part, that should be described in your results. But for your poster, you want to find a way to make that visual. Okay, sorry. So introductions, if you haven't already, I don't think people have submitted them yet, but that should be submitted by Friday to Moodle. And make sure that you are either addressing or incorporating your advisor edits. I did put a new assignment posting, so it's in a turn it in thing now, so that's going to be there. So if you've already submitted it, make sure you resubmit to the turn it in. And then next week again is a meeting day or working day. So we're not going to physically meet, but if you have questions that you want to bring up, we can treat that as an extra office hour. I'll just be around if you want for that. So you can be working on your methods and results. And we'll talk about that a little bit. And then also your poster draft is going to be due soon as well. All right, before I get back to the poster things here. So for your thesis in a nutshell, I remember this from 7th grade. This is what when we're writing essays, you guys remember doing like 5-paragraph essays. Your thesis is like that generally, except there's more paragraphs. So your introduction is, tell me what you were going to tell me, your results, methods, and if you're writing furiously, I will post these, so unless you want to add extra notes. So your results as methods is you telling me what you told me you were going to tell me, and then the discussion is tell me what you just told me. Okay. So in a final look, in your introduction, if you have not already done this, why is what you are going to tell me important? So if you're working with antibacterials, why do I care if you're going to make another antibacterial? There's lots of antibacterials around. Give me a reason why. If you're looking at drug combinations, like why is it important that we do this? If you're, whatever your project is, why is anybody going to care? Why does my grandma care if you're going to do something like that? Don't use my grandma and because she doesn't speak English and she doesn't drive and she doesn't do a lot of things. So she's probably not going to care about anything. So why should I care is the next thing. And then what has been done already. So give a brief, summary of what has been already done. And then what are you going to do? And then what is your research question, hypothesis, or objection? And if you want to state, my hypothesis is the objective of this project is this, go ahead and state it. But I need to know without having to guess, like, where is this paper going? What am I reading about? So try to include these things in your introduction. Hopefully your advisors have given you some input into this already. And then they have been either sending me the drafts that you've worked on or CC'd me on email responses to you if you're not one of my advisees, so that I can compare and see if you actually did it. So if you turned in your same draft to your advisor and to me on Moodle, you have not incorporated your changes, so that's going to result in penalties. So just make sure if there's something that you totally do not like what your advisor said, then you can address it on there, just in a note or something saying, I didn't like the way that this was worded, fine. But try to incorporate a lot of the changes or take their suggestions and incorporate those. Questions there? Anything not make sense here? All right. So then the results, the tell me section. I want to know what you did. How did you do it? So your experimental, if you're doing lab research projects, keep your experimentals brief. Remember, this is not an SOP. I don't need a stepwise procedure. And if you're not sure if it's long or short, find another paper in your peer-reviewed articles in your references that has a similar procedure and make it similar to that, swap things out. If you're doing a literature search or a paper, then what searches did you do? How did you refine things? How did you decide what papers to use and what not to use? So that's going to be your methods there. And then in your results also, it's going to be, what did you find out? In your lab projects, you'll have your results in tables and graphs. And literature, you'll have summary of your findings, which could also be in results of table and graphs as well. So if that is something that applies, so if you're comparing different antibacterials for whatever, you can have their activities or their IC50s or EC50s in a table. It's where your data is together. And I don't have to read through the results to figure out everything. Again, you guys like the visuals, try to think of a way to create a visual in your paper and on your poster. Because if you're doing it for your paper, you can just slap it onto your poster later. Okay, and then the discussion conclusion, what do your results mean? So you have to interpret them. If you did stuff with the ICP-MS, I don't know how to read ICP-MS data. It's weird to me. So you may have to talk to Dr. Wright about how you interpret that or what your data means. Maybe you know how to extract things from that. But your results section, if I go back to that, should not just be all of your raw data. You should be putting it into a meaningful table. Just like I would not have in my published paper listed all of the numbers of clones that I had to count. Like it's in a graph, okay, so it's summarized. So your results, you're gonna interpret those and figure out what that is. Same thing with your literature searches. And then for your conclusion, you're just gonna summarize everything and then tell me what you just told me. That kind of ties everything up in a nice package. Questions there? All right, so for your poster things, we liked, and you guys can go back to the discussion. I can add them to here or screenshot some things so that you have it in the PowerPoint that I post. But you liked visual aids, clean lines, probably bright pictures. We're not gonna knock the print quality because again, those posters are 2 years old. So they faded. The ones from last semester are out there. Those ones are fresher. So once we're also going to print you guys posters this year because we need to update the ones in the hall for prospective students. And it's just nice to have new research around. So those are things that you want to think about when you're working on your posters. Things that you did not like were but paragraphs, too many words, which is the same as paragraphs, bad quality pictures. So also keep those things in mind. So why do we make a poster? Scientists present their results to other scientists and also the public. So this is really, posters are actually really hard. Some people think, okay, when is the draft due? The 16th or something? I'll just wait till the 14th to do it. You would be surprised how much time it actually takes to make a poster. Once everything is on there, like futzing around is easy, doing aligning and stuff like that. That's super easy. But getting the information on there is difficult. Making a table to summarize your data is difficult. And then also choosing what to put on the poster even is also a challenge. So this is a really, it's hard. Posters you might think is easy, but it's actually hard. And then it's also hard to make it look nice, especially if you don't have something that is a lot of photographs. If you don't have nice pictures of gels to put on there, it can be challenging as well. Posters can be networking opportunities. So you'll see that. Okay, so I'm going to give you a minute to look at this and think of things that are wrong with this poster. I'm going to run and get my mouse because I need that.
I like the title.
And I was like,
Cancer
why is
drugs
this
and stuff.
person? And I was like, my eyelashes were wrong, they were white. Brendan was looking at white hair, because like parts of his hair was just like.
I aged very rapidly.
Yeah, I took a picture of that. I mean,
All right, what do we not like about this poster?
Too many words.
What else?
The picture is not one properly.
They're not aligned. What else is wrong with the pictures? No captions. It's a picture of a picture. So and some of these mistakes I do because I've seen them in other posters, but people will actually walk up to their computer screen, take a picture of the picture that they want to use, and then slap it on the poster. Yeah, so there's a lot of things wrong with this, right? No references current. Well, there's one.
For the whole thing.
Maybe it was, one. Okay, so a poster is not a place where you're going to dump your data. And then also some people like to just copy and paste sections of their paper, which is not okay to do because you guys don't like reading paragraphs. So we're going to put everything in bullet forms. And the poster is also not going to talk for you. Okay, so you want it to draw your people's attention. And this is one of the posters that I presented in Utah, and maybe it's scary because of all the different figures that you're not familiar with, but there's not a lot of paragraph. There's not a paragraph to be found here. It's all bullets. This is not a paragraph. This is the figure caption explaining all of these graphs. So if where you are right now, especially with literature projects, you probably won't have something like this where you don't have things, but it will draw people in, at least people who kind of know what you're talking about. So I'm probably not going to draw in a physical inorganic chemist to this poster because he sees things like biological data that is scary. But one of the things that I did with this one is pathways were a big part of the introduction of the project. Some people could put bullets out for all of these things, but isn't it easier to follow along if you see a visual of the pathway? So try to think about things like that. So you want to share the highlights of your story and then use it as a visual communication tool. Right. So the format of your posters, there are a couple of templates I know on the Google Drive that I shared with you, and then also on Moodle, that they should be 48 by 36. They can be landscape or portrait. So in biology, they like the portrait only because it fits more people into the areas. But whatever you think fits the way that your data is, you can do the tall or the wide. Use colors tastefully and with purpose. So somebody mentioned a bright yellow that they did not care for. This would also be really ugly. Try to keep your posters white and then use colors as accent, or if you have a color in your figure, that would be something to do as well. The flow, somebody mentioned poor flow. Usually it will flow, top to bottom and then left to right, depending on how your data goes. And you're not tied to a 3-column format or a 2-column format. Just make whatever fits your results best. We have the borders and then columns and edges there. You should have 1 inch between all of these things along there. And then we've already highlighted the minimal use of text and then using bullets. All right. So other things is you want to keep your tables and graphs simple. Number and figure, your figures and tables are as appropriate. So if your paper has 16 tables and you're using table 8 to put it on your poster, that should be table number 1 on your poster. So the numbering of them is not going to be the same as in your paper. It will be whatever it is on the poster. Skip the details of your study, like the fine details. You want just very broad things. So I had somebody doing peptoid synthesis and they had written out like every single painstaking detail. Nobody cares. Or if you're doing a synthesis, we don't need all of the reaction details and how many millimoles did you weigh out. It's just do the structure beginning, your reagents, and then the end. If they want the details really bad, they can ask you for it and you can send them a file later. Skip all of those minutiae of details and then use your pictures and colors to entice or draw the people into your poster. Titles. I noticed this with some of you with your titles of your thesis. Remember that your title should fit across the poster on 1 or 2 lines at 72 to 96 point font. So if you're finding that you have to shrink your title to 48 to fit, it's probably too long. And keep it interesting, but not cutesy. So try not to be, there was a kid in grad school who did a research talk on gold and he called it like a treasure trove of something and it was not cute. So try to stay away from cute, make it descriptive, it should fit. And hopefully it's not gonna be 125 point font because there's nothing else on your poster and you need the title to fit across there. So after the title, you have your name and LSSU, whatever it is, in a 48 point font. And these are also in the templates that you'll find on Moodle, the guidelines for the fonts. You have bold headings for your section, 28 point font for text and graphs and tables. You can use 24 for references and acknowledgement, but I would not go less than 24 unless you absolutely have to, because you do want to be able to read it standing 6 feet away and not have somebody doing this next to your poster. Questions there? Okay, so I talked about the format. There's the 2-column, the 3-column, and then you have the tall ones. You don't have to use any of these. Just figure out what fits your data better. And it may shift. You might start with a 2-column and find that you have to do 3, or it's not even columns. Leave some blank space, to let your columns breathe, but it doesn't have to be so much space that, again, you can tell that there's nothing, like there's not substance on the poster. So I just kind of did a Google image search of different poster templates, and you'll see that there's a couple of different types there. So let's see my mouse. This one looks like a 4-column format. One of the things that I hate seeing, if we won't have the poster session this year, and I'm gonna pick on not biology, but psychology, all of their students use the same 4-column format and the data doesn't fit in there. So you'll see introduction, methods, and then they'll have results that go into one and continue into the next column when the format should have just been different. So if you look at this one right here, we have, an introduction. You can start with your methodology and maybe your results fit better in this way or something like this, or maybe this is your whole results section. So don't feel like you have to fit all of your stuff into one template. Just kind of move things around and then see what works. So maybe this works better for you, just the 3 boxes and then 2 down here, and you have your acknowledgements and stuff like that, or something like this. So any questions on the template or format? So that's one of the things also is, and Caitlin Wilson, I worked with her last semester, and she had started one way, and then after I looked at the first draft, I was like, no. So then I made her, she had one of her flow charts going, I think, straight down or across or something, and I made her redraw the whole thing because it just didn't fit her data. And so that might come across, you may have to do that, which is why you save multiple drafts in case you have to go back to one of them. Okay. Your font, I don't care what font you use, be consistent. I know the default for PowerPoint is Calibri, which I personally hate, but you don't have to use my preferences for fonts. Limit to 1 to 2 fonts. So if you want to use something for the title that's different than the rest of it, that's fine, but don't use extra. And then make sure all of your text boxes are the same font after that, because I can tell. So a good title, you want to use something, these are serif fonts. Do you guys know the difference between serif and sans serif? So serif is like Times New Roman or Garamond, where they have these like little tails. I guess these are all serif fonts. And then sans serif is like Arial here, where they don't have the extra tails to the letters. So the serif ones tend to be easier to read. These ones are very difficult to read on posters, right? So I made the mistake of thinking I'm going to put fancy text on my checks, and I'm stuck with Old English checks. So use things that are easy to read, but that's going to be a personal preference. Limit to two fonts if you can. In your introduction, use bullets. I think we're all on board with that. It should concisely discuss what you're going to tell your audience. You can have pictures here, like I said, and also you can include a statement that says, this is my objective, this is the hypothesis. Questions here? Yes? Yeah, it's good to do that, like the first sentence or something like that, your first bullet point. That's very important, yep. And then this is where most of your references will be, unless you're doing a literature talk, is going to be in the introduction. The methods, it's going to be brief. Again, if you can make a scheme or some sort of flow chart, that's going to be better than listing a bunch of text. But if you have to do that, that's okay too. And captions for pictures go below. If you have tables, that goes above the things. And then the figures should go below I already said that. Results, clearly organized, summarize. Again, we're on board with the bullet points. If you can avoid text on your results, that's even better because your pictures say it all. But if you have to have text, that's completely fine. And again, tables go above, the titles go above the tables. All right, discussion, again, bullets, succinctly summarize your conclusions and what it means. And then you're not going to cite your figures and tables here. So you shouldn't have in your discussion, the conclusion was this as seen in figure whatever. That should be in your results section. And then it should relate to your research objectives. So if you said that your objective was to compare these two things, make sure you wrap up the comparison in your discussions and conclusions. And discussions, conclusion, if you want to put these two in the same box, the discussion conclusion, I'm okay with that as well. So again, it'll be how things fit for you. So this is not like a one size fits all. You kind of have to go based on what your research is telling you to do. References should definitely be included, but use them sparingly. To save space on references, one of the things that I tell people what you can do is to remove the journal article. So you have your authors and then like Journal of American Chemical Society, the year, volume, page number. So you don't need to include this is Bob's Backyard DNA Adducts, blah, 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 title part. So you can take that part out, and that usually saves you some space if you're short on space. All right, and again, ACS format. Acknowledgements that you should include. Whoever you want to acknowledge, but you use your good judgment there. So maybe not thank your dog, which some people do in biology. And then if you got funding, you should include that. Yeah.
So I went off of someone's research, I would acknowledge them.
Yes. Yep. Yeah. So if you have other students that contributed, you want to acknowledge them there. If you received URC funding, the undergraduate research funding, that should be included. If your research was funded by NIH or something, that should be included there as well. If you did not receive URC funding, that should not be on your poster. Or if there's other things, if you did not get, somebody did not pay for your research, then you should not include like the Natural Science Foundation, because you want to acknowledge them for fun. Okay, questions about poster format? All right, so we talked about some of these things. Use digital images of results where possible. Do not use cell phone screenshots. We already saw that in my ugly poster. Or take pictures of pictures. So after class, I'm going to put a poster outside of my office that kind of has a guide for, because I can't find the original digital file, but it has a guide for the pixels and then the percent sizes. So you can see if there was a picture that was like 600 DPI, what is that going to look like when I blow it up to poster size? You kind of have an idea of that, because some pictures you might find a nice flow chart on the internet, and then you want to use that, but the picture in real life is like this big, and then when you blow it up, it's going to look horrible. So that will just kind of show you what kind of resolution that you're looking for. We've talked about superscripts and subscripts, leaving spaces between values and the units, your degree sign. If you're using PowerPoint, the Office 360, which you guys all have access to if you didn't know that, there's always these guidelines and rulers to line things up, so use those. And if you can remake a table without spending hours and hours on it, that's going to be nicer looking than just copying and pasting a table from a picture. Okay, this is the PowerPoint tutorial. Oops. Portion of our talk, if I can get it here. Find that slide. Okay, how many of you know how to add a shape to PowerPoint? A couple of you, okay. So people in my class before have not known how to do this. So you insert, and there's different, where is it? Shapes. Whatever shape you want to do. If you're using a flow chart, you just draw the shape. You can change the color, the outline. One of the things you'll run into probably when you are trying to align shapes, if you have two shapes that are exactly the same width, and for some reason you can see in your eye that it's not aligning properly, it could be the border, which you'll have to figure out as well. So the shape, there's the fill and there's line. So if you had a border around there, which this one does, it's the width of the line that's going to be a little bit weird. So if one box had a 1.25 border and the other one had a 1, it's never going to line up like perfectly to your eye. So if you find yourself struggling with that, that's probably what it is. So if you are drawing, let's say this box here, and this is my text box, I don't want it to have color. There should be a no color option. Oops, that was the line. And then if you want to put another, text inside of here, you can just build another text box. This is very hard looking this way. A text box and just draw or just start typing right inside of there. So that's one of the ways to do shapes and text boxes, and you can't see it because something is behind. Oh, it's tiny, that's why. And white font. So you guys already know how to do font, and this font that I dislike. Okay, so whatever, me trying to type introduction, use spell check. Introduction, thank you. And then whatever size you end up wanting that to be, make sure it's highlighted. And then you have all those, the options of changing the sizes. So your text box can also be typed in there. And it's helpful, I think, to have the shape and then a text box inside of that, as opposed to just a text box, because you'll be able to move things around more easily, because there's a text box, and where's the other box? I don't know what happened to it, but you'll have a hard time moving things because this introduction wants to be this, however it fills the box. You can have it aligned in different ways where the text is on the top or the bottom. Those are things that you can change. So arrange, and there's different alignment tools here. All right, so for aligning things, this is the graph that I had, or the flow chart. And this is a lot of shapes that I drew onto PowerPoint that I had to move around. And let's say you need to move this a little bit, or the whole thing, and you want to move the whole thing. So what you can do is just drop your mouse and then highlight. You guys don't know about this, maybe? No? So I've highlighted everything, and you're going to group it so that wherever it needs to go, the whole thing is moving at the same time. Okay, and otherwise you're moving one thing. Let's say I decide to move this little line here, and then that throws everything off. But if you can group it, or once you have it done, you can ungroup it and do the same thing, or group half of it. So that's how you can move things around. Let's say, you know what I should be doing here? Make my life easier. All right, so I want to group or align some things. Stop. Ungroup. Let's say I move this little DNA thing somewhere, and I want to regroup all of these. I can just select the couple of things, and probably I've selected something I didn't want to. And then you can align here, center. So that aligned it, but this one didn't get clicked, so that one didn't get aligned. So it is a lot of futzing. Like I said, you would be surprised. See, I didn't want to do that. Surprised how long it takes to do these things. And if you have me for your advisor, I have extreme OCD issues. So you're going to be spending some time probably doing these things. But you'll have a very nice looking poster. So there's different things with aligning and grouping. You can, have things lined to the left, to the right, center, however you want to do it, top to bottom, all kinds of fun stuff. All right, here's some more things that you want to align. If you had your text boxes here, you want the discussion inside of that. Let's see if this is. All right, so this particular, I think this is one of the things in the template, but your text, if you just do a text box with a line around it, that is fine, but then it doesn't always grow the way that you want it to. So then this must have been an extra box. So you have to stretch it this way. So you kind of play with those things. You have your discussion borders. If you want to use these borders, you can. Just make sure it's aligned and inside. I can see an extra color on there. I don't like that. So this is the outline. If you want to get rid of that, just do no outline. And now there's not that light blue. Can you see the light blue here? So you won't have that. So just work on, it was just a lot of nitpicky things. Once you have your stuff on there, this is the stuff that you can be doing while you're watching Netflix, which will be I'm relaxed. I'm just going to do this like tiny work, but you have to get the heart of the stuff in there first before you can fiddle and watch Netflix while you're doing this. Okay. Your tables, if you have made a really nice table in Excel and then you just do the Control C, copy, and then Control V to paste it, it might go into PowerPoint like this, which I think personally is ugly, because this is what it looked like in the Excel, which I thought was nice. You had, things bolded here, bolded. One of the things you can do is explore the paste special. And then you can just paste it as a PNG or enhanced meta file, and it will paste it as a picture the way that you had it in Excel. So that's one way to get around that. Other questions on poster things? All right, so again, these are little nitpicky things that you pick up or you can do while it doesn't require a ton of attention span. You don't have to be completely focused. These are two posters that I dug up from Chemistry Archive from like 2004. And I was like, oh, this is a great, terrible poster because everybody said this, you don't like to read. And this is all, this is like a whole paper on the poster, which you want to avoid. This one is better because there are, nice pictures here, not necessarily probably having to do with the research, but this looks like an outdoor project. And so you have different graphs, there's some things there. I think this would have been, if we were gonna fix this, there's no references, but we would put everything in bullet points. One of the other things you want to avoid, even though this looks okay, is having a picture as your background, because that kind of draws away from your research there. Questions?
Also, would it be okay to have pictures that don't really belong in a section, so that those 4 pictures at the bottom, like they look nice, but they don't really belong to a section?
Yeah, so this is filler. This student is using it as filler. There wasn't enough to take up the space, so that's why they did that. If we have to do that, then we have to do that, but we prefer not to. You could make something bigger. I mean, I would rather have these be bigger and have the captions actually under it than to fill it with pretty looking pictures. Okay. This is another one that there was a lot of text on here and there's a lot of white space. I don't know if you noticed that in some of the pictures here, but there's just a lot of poorly used space. And like this is also very ill aligned, but you can see how, you know, the space is not used well there. This is a synthetic poster. We have bullets, there's a ton of structures. We have a chemical scheme giving the, you know, base results. This is an example of a good poster. And then there's a table here summarizing data. Everything is in bullet points. And this is another one, bullet points. These are, instead of doing structures, no, these are conditions for reactions. Again, this is a lot of work. When you see something like this, people are trying to cram everything they did into a poster, which is fine, but it can be overwhelming. Some of you may be scared off because you don't like organic chemistry, but that's what that one looks like. And this is one where there's still bullet points. And if you look, the formats were different for each of these too. So this is a person that did a tall poster. They have, you know, the results section in the middle with nice colored images that are big enough to be able to read. And then they have just everything bullet pointed there. There's pretty good use of space there, but that's the best we could do for that one. All right, questions on your posters or introductions or anything else senior seminar related? Yes.
Okay, this might be a stupid question, but like how do you start, like what do you use to make the poster?
Oh, there is, if you go to Moodle, there's a tab that I think is templates and examples. So you can just you can start with the PowerPoint template that's already there and build off of that one. But I use PowerPoint to do all of the posters.
So then like the size of the like font sizes that you were saying, like those line up with PowerPoint?
Yes, yeah, so
one slide would be your whole poster? Is that what you're saying?
yes, exactly. So yeah, your whole poster should not be 6 slides. All right, let's go to Moodle quick. Oh, you guys can't see that. All right, so templates and examples. I just have a couple of basic ones here. So there's the 4-column PowerPoint, so you don't have to change the size of the poster at all. And then you have the 2-column, there's a couple of ones in there. There's I think these were the ones that I put on the slides, but your examples of good and bad posters. And then any of the graphics that you want to use, I have those here as well. If you guys like the new chemistry flask ones, those are on there. And there's specific ones also. So there's chemistry, there's a cannabis flask, there's a biochemistry one, and then a forensic one, if you want to put those on your poster. But the base template is there, so you have that available to you. Other questions? All right, so the last slide was just a reminder of what's due. You have your introductions due, and I'll try to get your feedback on that as quickly as I can. It does take some time to read through those. Make sure you're uploading a Word document so that I can do track changes in there. That's a lot easier to edit than trying to scribble stuff in between. No class next week, but I'll be around if you need to find me, your methods and results, and then work on your posters. Questions? All right, I will see you in a few weeks. Stay warm out there.
time would you like me to come in?
Whatever, when do you have your class?
Just 9 to 10:30. So,
I'll be in lab probably. I might be
Okay.
done by that point, so you can just come in after class.
okay.
And then, so you're just swabbing
Yeah,
and then getting your, you have money from one place, right?
I got 3 places.
Oh, you got-
I
okay.
them all. So,
Awesome. That's a lot of cash. Yeah, so you can just do the,
$160.
the swabbing and then, yeah, then we can get your samples ready.
Okay.
Were we gonna run that on the
LCMS.
LCMS? Okay.
All right, sounds good.
How frequent